the purpose of my talk is really to talk about what are dry surface biofilms, the evidence we've got to date about those dry surface biofilms, and of course, for the purpose of this meeting, is the implication for the food processing environment. So I will start with a slide about biofilms, and obviously you've probably heard a lot of biofilm today already. When we think about biofilm, we're thinking more about now what we can call a hydrated biofilms or a wet biofilm. So you've got in your mouth what you've got, but for the food industry, it's more what you've got probably in pipes, some part of the machinery that cannot be cleaned very easily. On that might include conveyor belt as well, where on that pictures here, you've got some biofilm here growing that. And those are the more traditional biofilms that probably people think about rather than some biofilm which are on dry environmental surfaces. So the field of dry biofilm is quite a new one. On the term dry surface biofilm was coined by a colleague of mine from Macquarie University, Karen Vickery, and she's the first one really to identify those dry surface biofilms quite a number of years ago. And the first publication on that was in 2012. And since then, there's been a bit more publication there and then, all on the healthcare environment, and that's the primary evidence we've got, because we're starting to look at those biofilms in healthcare. So we certainly joined Karen's on looking at those evidences of dry surface biofilms in healthcare, and I'll talk a bit more about that in terms of evidence. And so we've got quite a number of articles referring to dry surface biofilm. On this one, I also refer to during my talk from a colleague of mine, Michel Alpha from Winnipeg, and on other scopes as well, and we'll have some talks there. So quite a few publications over the years, but overall, we've got about only 15, 16 publications only. So it's quite a new field of avenue for, I guess, biofilm research. So, and it's now moving on to how difficult it is to remove from surfaces, the effect of cleaning and disinfections on the dry surfaces, on those dry biofilms, but also the impact of transmissions of pathogen from those dry surface biofilms. So what are dry surface biofilms? Well, there are complex microbial communities formed and grown in dry habitats and on surfaces. And evidence show that they can colonize various materials from textiles, but also from hard surfaces, including plastic, wood, metals, and so on. The importance of those dry surface biofilm is you can't see them. They are only very small. They're only few microns. So you might imagine that, or you might think that the surface is free of microorganisms, when indeed it does contain some dry biofilms. And I will talk about how difficult it is actually to look at those dry biofilms in a minute. So we've really isolated those dry surface biofilm from a various environment, but what they've got in common is low moisture, various temperatures, depending when they are, and of course, various nutrient levels, usually debris, food debris, and so on, deposited on those environmental surfaces. So let me tell you about how do we identify those dry surface biofilms, and I'm afraid that, yes, it will refer to healthcare environment, but that's an example of a study we've done between 3 hospitals in the UK, collecting samples that can be sanitizing bottles, keyboard, patient folders, and clipboards. In that study, in particular, we also look at a bed as well for another study. And what we've done with those surfaces is looking for dry surface biofilm, the evidence of that, abundance and diversity of microorganisms using next generation sequencing, and also the appearance with scanning electron microscopy. But on that, studies reported in that publications a few years back. But really, for you to understand really what we're talking about is, first of all, the samples. The way we look at dry surface biofilm is the destructive way for the samples. And so when we get our material from the hospitals, very often they are being decontaminated with sodium hypochlorite to start with. Then we suddenly with the sample we rinse the samples with sterile water, and then when they dry we swab using like a cotton swab that is commonly used in healthcare settings to look at microbial contaminations. So we're using swabbing, wet swabbing on the surfaces, on grow the swabs on agar. And when we've got no growth, then what we do is we cut those surfaces in very specific size, and we use an enrichment broth. And when we've got growth, then we know we've got some microorganism in those surfaces that were not picked up by the swab. And then we go back to the surface and looking for imaging as well, or we're looking at adjacent surfaces from a sample to look at imaging as well. And with the broth, we're looking at NGS sequencing, but also we use selective media. So some results here that depending on the hospitals, and yes, we found loads of microorganisms on surfaces. And again, those surfaces were free of microorganism when we looked at swabbing. But we identify those microorganisms on the surfaces. On all the surfaces, we identify by scanning electron microscopy those dry surface biofilms. So again, they were not picked up by swabbing. And in two hospitals, we've got very similar pictures where you've got the gram-positive, a staphylococci. Mostly, one hospital was slightly different. We had lots of bacillus, again gram-positive, and that was quite surprising. But anyway, the end of the story is we found loads of microorganisms on surfaces. So one of the conclusions is in 95% of the surfaces we sampled, we find those dry surface biofilms. So they're very widespread. All of them are gram-positive predominantly. We find some evidence of DNA of gram-negative bacteria as well, but by far that was minority. And also part of what we're looking to do with those dry surface biofilm is their ability to regrow, especially when we get the chance of a rich environment, especially nutrient. So all the surfaces were able to really nicely grow a microorganism when we thought they were free of microorganism to start with. But the most important point of all is those dry biofilms could not be detected by wet swabbing. And that's normal cotton swabbing. We're not talking about a flaked swab as introduced by Rob earlier on this morning. So lots of questions as well is how widespread are those dry surface biofilms? And on the story of dry surface biofilm really started with breast implant with Karen Vickery in Macquarie University, where with quality control, a microorganism could not be found on those breast implants, especially the RAF breast implant, which are much better. But there were issues when implanted because they had some contaminations, and they found actually some microorganism associated with those breast implants as a dry biofilms. There's also questions about a medical instrument, especially endoscopes. And Michel Alpha, I mentioned just 5 minutes ago, had done a fantastic review about really looking and trying to understanding what sort of biofilm do we find on those equipment. And she really in the lab developed a method to look what's called build-up biofilms. On BBF, they call that. And there was very lots of similarity between those BBF and those dry surface biofilms. One of the big difference in methodology is when we grow dry biofilms, you will see that at the end of this presentation, is we don't add microorganism on surfaces repeatedly. We just start with one inoculum, which is a bit different from Michel's protocols. But at the end of the day, the appearance of those microorganisms on surfaces, especially surfaces following treatment with disinfectant or sterilant, are very similar between those dry surface biofilms on what she called the build-up biofilms. Very similar, very thin, scattered randomly on the material we observed, on at the end with the presence of EPS as well. So knowing that we've got now those dry surface biofilms very widespread in healthcare environment, that we've got evidence that whether or not you count the term dry surface biofilms or build-up biofilms on medical devices, is what is the implication for the food processing environment? I mean, are they going to be an issue? Can we perceive them as being an issue? And how about the effectiveness of disinfection against those dry surface biofilms? So again, when we compare the two environments, then we know now with healthcare that the dry surface biofilm can be responsible for persistence of pathogen in the environment. They cannot be detected from the dry surfaces using normal wet cotton swabbing. We know as well they are less responsive to biocidal product. I will show you some evidence at the end of this presentation. And then there's the remaining questions about are they involved in recurrent infections? And by that I mean is if you clean a room in the hospitals where you had a patient with MDRO, if you deep clean that room, then the next naive patients using that room got a very good chance of getting that MDRO. And nobody knows where it's come from at the moment. There's few theories, but we believe it's because of those dry surface biofilms. So of course that's got an impact on hospital hygiene, on ultimately patient safety and staff safety. Now, if we turn our attentions to the food industry, the question is, can dry surface biofilm be responsible for the persistence of pathogens, of food pathogen surfaces? Can they be responsible for food contaminations, decrease food performance in factories, especially in manufacturing food? Can they be involved in decreased shelf life of the product? Obviously, at the end, customers trust. And those questions will need to be obviously answered at some stage. So again, when we really focusing on the food industry, I mean, we've got some evidence that obviously foodborne illness are very important. I think we had some brilliant presentation this morning on that. We know that a pathogen can be transferred between food surfaces as well. That's very well published. We know we've got plenty of biofilms, and again, I will refer to the excellent talk from Rob this morning that really show the extent of which you've got very complex biofilms in different parts of or different food manufacturing facilities. And whatever facilities you're looking, whether it's mostly on fish or dairy or meat processing, then you will find those biofilms on various equipment. That is very well exemplified. But in most of those studies, we're really looking at more to a wet biofilms rather than those dry surface biofilms. We know the consequences, obviously. I'm preaching the converted already, that again, if you've got any contaminations, food contamination of the product, then the consequences of can be very severe for the companies, but also obviously for the consumers as well. And the question is really, could dry surface biofilm be implicated in those? And well, as you will probably see in a minute, that, well, yes, we know that dry surface biofilm can survive a very long time on dry environmental surfaces. We know that many treatments at the moment, from what we know, fail to eradicate those dry surface biofilm, highlighting how resistant they are to a normal cleaning and disinfection procedures. And we know they are transferable in certain conditions as well. So again, all knowledge we have from the healthcare industries is applicable to the food industry, certainly. And we do hypothesize that dry surface biofilm, as I said, may be the source of recurrent healthcare-associated infection in the healthcare settings, but they may equally play a role in product contamination in the food industry. And again, to reiterate, I mean, some of the talks we've seen this morning is in different part of the food industry, whether it's the meat and poultry industry, the fish and seafood industry, or the dairies, then we know we find multiple microorganisms in biofilms. And there's been quite a very number of studies here. I quote only three of them, looking at different environment. And the first one was looking particularly at conveyor belts and what type of microorganism or complex biofilm they're finding on those conveyor belts. The same with that study was on salmon as well, or the fish on seafood industry, on looking at what they found after the equipment was sanitized. The surface on the equipment was sanitized, so they still find microorganism lurking there. On us for the last one on the dairy industry, that study in particular, looking at contamination of a reverse osmosis filters on ultrafiltrations. But at the end of the day, all those studies found those microorganisms, or example of studies found those microorganisms in different food environment. I would like to highlight also that excellent review from Moretro on Langsgren as well, with looking at the literatures, looking at what can be found in different industries, on again, both gram negative and gram positive here obviously are present in various food industry, and depending on the industry, you're going to have more species than others. But when we look in closely at all those papers that have been reviewed in that excellent piece of work, is it is clear that no dry surface biofilm were analyzed, because again that's a new subject, and almost all studies use a wet swabbing or contact plates. On that point, it was quite interesting to see again the talk by Rob this morning, looking at there was a special a swab they were using to scratch the surface, to trying to get more microorganism there. On that's quite interesting as well. But also lots of studies that in that reviews analyze very few samples, and many studies also used a selective plate instead of a sequencing. And again, that thank you to Rob this morning is that talk distinguishing between what you get if you use selective media versus if you're looking at a more robust sequencing. You get far more microorganism on the two a method between NGS, for example, on the selective plate might not matched up. So overall, plenty of evidence. You've got microorganisms in the food industry persisting on the surfaces, found regularly, but we don't really have looked at the presence of those dry surface biofilms in this case. So as a sort of the conclusion of this part of the talk is again, plenty of evidence there. You've got microorganisms and complex community of microorganisms. We don't know if you've got dry surface biofilms. The compositions, the complex compositions, is starting to become really understood using those, I guess, sequencing methodology as well. And of course, one of the issue is specifically for the food industry, what the cleaning and disinfection regimen used then are not really being tested. All the evidence I'm going to present to you in the next few slides is a product that mostly used obviously in healthcare settings. So really, we do recognize, whether it's in the food industry or the healthcare industry, is at least that current cleaning practices or on disinfection regimen are not adequate really to control biofilms in the environment, and that's the biggest challenge we have. In the food industry, it's only the variety of biofilms, depending on the environment as well, is extremely challenging to provide an efficacious decontamination procedures for those type of microorganisms in biofilms. So really to understand better what dry surface biofilms are, the characteristics, on especially the susceptibility to cleaning or disinfection regimen, then we need to produce them in the lab. You've got to know the choice, because what you've got in the environment, they are very complex and a bit random. So we've done in our lab a a system to culture dry surface biofilms, usually monospecies. Now we're doing dual species as well. It takes 12 days. And the way it works is a sequence of wet and dry phase over a 12 days period. And at the end of the 12 days, you've got those dry surface biofilm on the surface. The characteristic of the surfaces is the surfaces is nicely covered with those dry surface biofilms. And we've used that method to produce a dry surface biofilm with a number of species. We started with Staph aureus, but use also Candida auris, Bacillus subtilis, and then environmental Bacillus licheniformis. Also use with that with Klebsiella pneumoniae, but also with some food pathogens, Salmonella enterica, and Listeria monocytogenes that do form very nice dry surface biofilms. And because we're able to reproduce a nice dry surface biofilms, then we can then test solutions to combat and understand the efficacy of intervention against those dry surface biofilms. So really briefly about a food-related pathogens. On the left-hand side, that graph is after 12 days growing those dry biofilms with a Listeria and Salmonella. We've got on top of our surface about 10 to the 8 CFU per in this case, a ml, CFU per coupons that we've got on the surfaces. Very similar to other microorganisms we've used before, but of interest, certainly for the purpose of this talk and the relevance to the food industry, is our dry biofilm with Listeria monocytogenes do survive very well in their environment. Here, the environment was 20 degrees after 50% relative humidity, and you can see that, even after 8 weeks, then we still got about 10 to the 6 of those dry surface biofilm on surfaces. So again, that provides us some evidence that in the form of a dry surface biofilm, those pathogens can survive quite happily in the environment. So this is what they look like. I mean, that's a Staph aureus dry surface biofilms. That's from on the left-hand side here. It's from our artificial biofilm we're growing in the lab. So you can see those Staph aureus, they're a bit stressed. They're not really looking that well, but you've got evidence also of lipopolysaccharide. And that's compared with some dry environmental biofilms we find. And you can see that, again, there's gram-positive here. Really quite similar, except on pictures we've got in both cases. We also looked at viability of those dry surface biofilms. We grew our dry surface biofilm in the presence of organic load. It gave us better viability after 12 days, on certainly help in the cohesions of the biofilms. And again, we reported that in that paper here. But most importantly is how do we test for the efficacy of product. In healthcare, obviously, pre-wetted wipes are heavily used. So we've used, obviously, a system that enables us to use pre-wetted wipes against those dry surface biofilms. But what is very different, the way we do things now is not only we're looking at the reductions in microorganisms that are being removed or killed on surfaces by those wipes, but we're also looking at transferability on regrowth, so the ability of the biofilms, the dry surface biofilm, to regrow. Transferability is also extremely important. Obviously, we want to make sure that the surface is safe after the cleaning or disinfection regime. You don't want to have to transfer microorganisms between the surfaces after they clean. And what we've said is in those two papers, one is in press at the moment, is obviously for us really a good product now is not really measured just because it's nicely killing microorganism, but it's also because it's transferring or it's decreasing the transfer of microorganism from surfaces after the product has been used. So here are some results of that paper that is in press at the moment. On this side here, you see the interventions. I mean, some of those would be used in the food industry. Here we've got benzalkonium chloride. We've got different chlorine compounds. Here it's peracetic acid, chlorine dioxide. Here, interestingly, it's a vaporized hydrogen peroxide, which is extremely powerful way to destroy microorganisms, and usually used in healthcare when you've got an outbreak that is difficult to control. You would be interesting here with the a cold plasma. We've tried those dry biofilms, and here there's a control. Briefly, again, three indicators. Three indicators: the reductions in viability, the transferability, which is direct transfer, looking at the surfaces, and by adpression to a growth media, we're looking how many microorganisms can be transferred. And then for the healthcare industry, really the regrowth. I mean, can those biofilm regrow, or can those interventions stop those biofilm to regrow after a number of days? So you can see those results. Then if we're looking at the log reduction only, you can see some interventions were pretty good, but quite interesting. Again, those dry surface biofilm, you can see that vaporized hydrogen peroxide, which is extremely powerful, had no activity with those parameters. Neither did the cold plasma. But when we're looking at a direct transferability, then you've got starting having a better picture where the intervention with the benzyl alkylum chloride formulation was much better. The same with peracetic acid, but if we're looking at a sodium hypochlorite, then suddenly not as good. And when we're looking at regrowth, which is the latest indicator we look at, is only one product out of all of those actually provided quite a decent information as how robust that product is. Now, in terms of other transferability, we were very interesting about contacts with fingers as well. And here we're looking at gloves and different type of gloves in the transferability of those a dry surface biofilm after in interventions. And again, what you can see here is quite a different pictures. I mean, those I guess breakpoint less than 2 log and more than 2 log. Again, they are what we think is reasonable, what to expect from a product in terms of transferability. And you can see, again, quite a mixed pictures between different products. Some of them work with one type of gloves, but certainly not with the others. Overall, I mean, what that tells us is those dry surface biofilms are quite resilient to interventions, whether it is with a detergent or whether it is with a disinfectant. On looking just at the reductions of those microorganisms on surfaces is certainly not enough to get better understanding of how the product works and how efficacious the product is. So really what we have in terms of problem is we do have transferability and a recovery of dry surface biofilm post-treatment. We know that most disinfectant cannot prevent the transfer of DSB between surfaces. And we know that, as I just shown you, that you can have a very high efficacy in terms of reducing the viability of those microorganisms, but it doesn't always correlate to the performance overall of the product. So going back to our implication for the food industry is, well, healthcare, we know they're widespread, they are persistent, they're not easily detectable. We can recover them on the pathogens in a dry surface biofilm can be transferred following certain conditions, especially when you use a detergent on the surface is very wet. But for the food industry, well, so far there's been no study on dry surface biofilm in the food industry. Well, I think if we start looking for them, then we will, I'm pretty certain that we will find those microorganisms there. So that's the end of my presentation. I'd like to thank you for listening. I would like to thank my postdoc, Katarzyna Ledwork, who's presenting part of his presentation at IFP a couple of years ago. So I sort of steal a slide, and quite a number of people as well who've been involved in the story of dry surface biofilms for the last probably 6 years, been working on that. So thank you very much indeed.
Jean-Yves, thank you very much for a fascinating talk and a really helpful description of this whole emerging issue of dry surface biofilms and how that might relate to the food environment. So I'll look to my colleague Will to just ask, are there any questions from the audience that we wanted to pose Jean-Yves at this time? We've got time for a couple of questions.
Absolutely. So the first one comes from Steve F, and he asks, do you think that the source of dry biofilms is due to aerosols which are easily dispersed by airflow, then settle on a surface and then dry, which would mean that wet cleaning could be a contributing factor?
I think for the food industry, I can't say, but for the healthcare industry, where we find them is a mostly fomite contact, food residues, a hand contact, a object contact as well, probably rather than aerosols. But what is clear is for us is when you've got those microorganisms on the surfaces, desiccation of those microorganisms on surfaces, and then rehydration of those microorganisms because you had more food, because you had some cleaning with water and so on, might contribute to the formation of the biofilm. But we're actually looking at that at the moment.
Great, thank you. Next one is from Michelle, who asks, what makes these cells an actual biofilm and not simply a discrete dormant contaminant on a surface?
I mean, that's good questions on this one, and we certainly thought about that. But what we found actually is you've got, against all the characteristic of the biofilms, especially in terms of compositions, in terms of carbohydrates, proteins, compositions, DNA, but also in terms of exopolysaccharide evidence in those dry surface biofilms. I mean, I would go to say in terms of distinction between dry surface biofilm and wet biofilms, in those dry surface biofilm, there's no much evidence of extracellular DNA in the matrix. It's mostly protein and carbohydrate, EPS, produced by those cells.
Thank you very much. If we go for the next one, we've got, does your dry surface biofilm formation technique promote sporulation?
It would in a certain way because you're stressing the microorganism. So for those spore formers, especially the bacillus, and within the dry surface biofilm, within that matrix, when we're making we've got some spores there, but we've got the majority is still vegetative cells. So when we testing, for example, the efficacy of product, and especially sporicides, against those dry surface biofilms, then the ability of those bacillus to survive is not only explained by the presence of spores, but because they're stressed, then yes, you will have a mix of vegetative cells, but as well spores with the bacillus species.
Thanks very much. I'll do one quick one now before we get to the end of this time slot. We get one come from Jeff Banks, who asks, do you know or would you suspect that the chemical and physical composition of the food matrix could play a more significant role in both microbial persistence and microbial shedding within a dry biofilm when compared to a wet biofilm?
Absolutely. I mean, if we're looking at the certainly food industry, I mean, the way you're, I mean, with the first talk with Karen, it was clear that cleaning and disinfection was so important. So you're expecting a very good hygiene, but then you're going to have some food debris there from time to time, which might be very difficult to clean in some species and so on. So that will certainly, in my mind, contribute to certainly helping the dry biofilm to grow. But then again, if you've got too much food and too much water, then you're going to have sort of a wet biofilm. You can see that was very nicely exemplified by Rob this morning. And as I said, certainly we'll talk a bit more to Rob, because I mean, using those scrapers for this experiment he's explaining this morning, I just wonder if he's also managed to get some possible dry surface biofilm in this case. I've got to say, we've never used those flecked swab on our surfaces to look whether or not we can catch those dry surface biofilm.